Hello, Assalamualaikum and warahmatullahi wabarakatuh and very good morning. We from Group 4 including me, Hazim, Tanesh, JV and Faris, we introduce about cytometry. So what actually are cytometry? Cytometry is a technique used to analyze the physical and chemical characteristic particle, typical cell, as they flow and fluid stream through a beam of light. So, cytometry machines are sophisticated instruments designed for high throughput analysis of cells, enabling researchers to dive into the intricacies of cellular characteristics. So, what a function of cytometry? Firstly, is for cell analysis. Flow cytometry allows for the analysis of cell based on properties such as size, granularity, and fluorescence. Cells are suspended in fluid and pass through of laser beam, and detector measure the scattered and emitted light. Fluorescence detection. Cell can be labeled with fluorescence dye or antibody that bind to specific cellular component. This allows researchers to examine the presence of in situ of fluorescence, providing information about expression of protein or other biomolecule within cells. Third is cell counting. Flow cytometry provides accurate cell counting, help researchers determine the concentration of cell in a sample. Fourth is cell cycle analysis. Flow cytometry can be used to analyze distribution of cell in different phases of the cell cycle, providing insight into cell proliferation. And five is immunophenotyping. This technique is commonly used in immunology to identify the characteristic cell population based on their surface marker. It helps researchers distinguish between different cell types within heterogeneous population. And lastly is hypothesis detection. Flow cytometry can be employed to assess apoptotic process in cell by measuring changes in cell size, membrane permeability, and DNA content. That's all for me. So I pass to Tanesh.
Principles of cytometry. Cytometry is a combination of two words, where cyto means cells and metry means measurement. It is basically a technique of measuring cells. The basic principle of flow cytometry is the passage of cells in a single file in front of a laser so that they can be detected, counted, and sorted. Cell components are fluorescently labeled and then excited by the laser to emit light at varying wavelengths. The fluorescence can then be measured to determine the amount of types of cells present in a sample.
The component of cytometer is divided into three parts. The first one is the fluidic, followed by optical and electronic system. The fluidic system of a flow cytometer is responsible for transporting sample from the sample tube to the flow cell, while the component of the optical system include the excitation like sources, lenses, and filter used to collect and move light around the instrument, and the detection system that generate the photocurrent. The electronic are the brain of the cytometer. Here, the photocurrent from the detector is digitized and processed to be saved for subsequent analysis. The purpose of the fluidic system is to transport particle in a fluid stream to the laser beam, and the design of the flow chamber allow the sample core to be focused on the center of the sheath fluid, where the laser beam then interact with the particle. And this is achieved by applying the hydrodynamic focusing, where a fluid was used to surround the sample fluid. And this surrounding fluid is known as the sheath fluid, where it flow at a very high rate. By injecting the sample into the middle of the sheath fluid, the differences in the velocity and the density of the two liquid allow the cell to flow through the tube separately with minimal mixing, and the cells are forced to pass in a single line through the optical laser beam. And with cytometer, thousands of cells can be analyzed and sorted per second. But what allow for this precise measurement at such high speed? The first part comes from the optic system. The optic system consists of the laser and lenses that are used to shape and focus the laser beam to the flow of the sample. And the place where the cell interact with laser light is called the interrogation point. You might also hear it referred to as the laser intercept. And this is where the action take place. When the laser light beam illuminate a single cell, some of the light will strike physical structure within the cell, causing the light to scatter. And this light scatter can be measured and correlate with the relative cell size and structure inside the cell. There are two types of scatter in the optic system, where the first one is the forward scatter, where the light will scatter past the cell. The light scattering past the cell typically identify the relative cell size. The intensity measured on the light scatter detector correlate with the diameter of our sample. For example, to differentiating between white blood cell is one area where forward scatter play a role. The second one is the side scatter, where light that is scattered perpendicular or 90 degrees is filtered through a number of lenses and mirror. Where forward scatter measure the diameter and the volume of the cell, side scatter help to provide information on the complexity of the cell. And side scatter can be used to determine the nuclear structure and the granularity or the texture of the cell. And combining these two detection methods allow for a great deal of differentiation between cells. The last part is the electronic system where the detector converts the fluorescent signal into a digital trackable data point. And this data point then create images that can be analyzed to measure the different cell apparent in the heterogeneous fluid. And these are some of the example of result that is produced by cytometer. It combines the two scatter method, the side and forward, to differentiate between the two type to the white blood cell.
Cytometer can be used to do qualitative and quantitative analysis. It can provide rapid analysis of multiple characteristics of a single sample. Modern cytometers are easier to use and is less expensive, which is why they have wide range of application. For example, DNA content analysis. By using fluorescent dye, cytometry can measure the content of DNA and detect any changes in the DNA. This could help detect abnormality in the DNA that could be related to disease. Next is immunophenotyping in hematology. Hematology is the study of blood and blood disease. So cytometry can be used in hematology, more specifically in blood bank, usually to detect leukocyte contamination in blood product. Another example of the application is for chromosome analysis. So chromosome analysis is a time-consuming process that requires a faster and more efficient alternative. Cytometry can provide a high-speed quantitative karyotyping that overcome this time constraint of traditional chromosome analysis and allowing for more chromosome to be analyzed in order to identify disease. So these are just some of the example of the application. There are way more which cannot all be mentioned in this opportunity. Thank you.